Another control point for gene expression is during translation. And how this happens is through regulating the mRNA that has been established from transcription. There are two special types of RNA that are able to halt translation: microRNA and siRNA, small interfering RNA. So their process is similar, but then a little bit different. They both bind to the mRNA strand once it's in the cytosol or the cytoplasm. We have to remember that when that mRNA is able to leave the nucleus, it is going to be translated at the ribosome, and that polypeptide is built. So by the microRNA and the small interfering RNA binding to that mRNA molecule, it essentially blocks it from being translated. Now, where they're different, microRNAs degrade the mRNA strand, so they break it apart and make it not useful. That's what degrading it is. Now, the only way that it can be degraded is if the code, the bases match up perfectly. Then it will be degraded. With the small interfering RNA, it has a protein, it has the special RNA strand, and it's not able to go into the ribosome for translation. Here's a diagram summarizing the last slide, putting a visual together with the microRNA, how it ends up degrading the mRNA that has been transcribed from transcription. So up at the top, you see the purple blob. This is going to be a protein. And then inside that protein, you have the microRNA. So it's the microRNA protein complex. So it is going to seek out the red strand, which is the mRNA, that is looking for a ribosome for translation to occur. So it runs into the mRNA, and if you look on the left, the purple blob with the gold the microRNA along with the mRNA strand, which is red, you can see, if you look really closely, there's little lines showing that the microRNA and the mRNA bases are completely complementary, so they match up in that complementary fashion, like A's and U's and C's and G's. So when they completely match up, the mRNA is going to be degraded by that protein or that enzyme, essentially. On the right-hand side, if you look very closely, you can see that not all of the bases match up. So in this case, they're going to stay stuck together. The mRNA is not going to be degraded, but because it stays all connected to one another, translation still cannot occur. It can't make its way to that brownish blob at the bottom, which is the ribosome. So in both of these cases, whether the mRNA gets degraded or the translation is actually blocked, which they both basically block translation because we can't in either case build that polypeptide. This is another way that gene expression is controlled, by not allowing that polypeptide to be built. These are the highlights of section 15.2 and 15.3. So please make sure that you understand all the highlights of every control point, starting with chromatin all the way down to the protein being built, all the different checkpoints for gene expression, how it's regulated.